Hello viewers, in previous lecture we discussed about organochlorine pesticides. Under this category we discussed briefly the synthesis, the effects and the uses of DDT and lindane. In this lecture we will discuss organophosphorus pesticides, then their synthesis, uses and adverse effect of parathane and malathion. Organophosphate pesticides. These were discovered and synthesized by German scientists in 1930s, and parathane was one of the earliest organophosphorus insecticide used. Today more than 100 organophosphorus pesticides are used in countries such as Thailand, Vietnam and India for public health purposes and to protect crops. Organophosphorus compounds become the leading type of insecticide as they are less persistent. Gerhard Schrader synthesized the first organophosphorus insecticide in Germany in 1938. It was commonly called TEPP, tetraethyl pyrophosphate. It is probably the most toxic substance ever used in agriculture. These are some examples of organophosphate insecticides. You can see the chemical structure of ethane and malathion. This is parathane and this is phenthine. Let us concentrate on parathane. This is the chemical structure of parathane. The formula is C10H14NO5PS. Its name is OO-diethyl O,4-nitrophenyl phosphorothiate, or it is also known as ethyl parathione or parathine ethyl. Ethyl parathine first registered in 1948. It was once a restricted use insecticide with limited applications only on certain agriculture crops, but by 2003 all registered uses were voluntarily cancelled. Parathine is one of the most toxic pesticide. Persons whose skin or clothing is contaminated with liquid or powdered parathine can cause secondary contamination by direct contact. Parathine was synthesized by Schrader in 1944. There were two methods for its industrial synthesis, which differ only in preparation of the OO-diethyl phosphorothiochloridate. The German process starts with thiophosphoryl chloride, but the American with phosphorus pentasulfide. Only this is the difference, while the last step it is common. This is phosphorylation of the para-nitrophenol sodium salt is the same in both routes. Numerous variations are suggested for the special reactions condition. Let us discuss the synthesis of parathine. Thiophosphoryl chloride PSCl3 was treated with sodium ethoxide in first step to form OO-diethyl chlorothiaphosphate. Its formula is C2H5O whole square PSCl3. In next step, this was heated with sodium para-nitrobenzene solution, which gives the product parathine. Let us discuss the properties of parathine. At room temperature, it is a combustible pale yellow to dark brown liquid that may be difficult to ignite. In commercial products, parathine is usually dissolved in hydrocarbon solvents such as toluene or xylene, which are flammable, and weak odor of garlic from this parathine insecticide comes. Parathine is a broad spectrum insecticide which acts as a contact, feed or respiratory poison. It is widely used against biting and sucking insects in agriculture, for fruits, in vegetables and vine growing, as well as against mites, beetles and caterpillars. It is usually applied as a spray in emulsion form 500 gram per liter, and in that oil is 10%. Parathane is only moderately persistent in the environment, that is the good point, and it does not accumulate in the biosphere. On the other hand, it lacks sensitivity and it is highly toxic to bees and beneficial parasites, fishes, mammals and humans. Parathane is interesting because it does not show selective toxicity between animals. It is strong poison for both sexes and all stages of insects and vertebrate life, and its exposure through skin or through the mouth is equally effective. Parathane has a high vapor pressure and can poison insects with vertebrates through the respiratory system. Let us discuss the points of attacks. Parathane attacks on respiratory systems, lungs, skin, eyes, on plasma, cardiovascular system also, central nervous system and red blood cell cholinesterase. Let us discuss the harmful effects of parathane. Systemic toxicity due to parathane can result from all routes of exposure, and it can cause abdominal cramps, vomiting, diarrhea, blurred vision, excessive sweating, salivation, wheezing, excessive trachebronchial secretion, agitation, seizures, muscle twitching, weakness and urinary bladder incontinence also. And the heart rate may decrease following oral exposure or increase following dermal exposure, and chest pain may be noted. And the seizures are most common in children than in adults. Shortness of breath may be followed by the respiratory collapse, giddiness is common, and some symptoms are it is extremely toxic material. The probable oral lethal dose is 5 to 50 milligram per kg. As little as 1 drop can endanger life if splashed in the eye. So, how much it is toxic you can understand. It is cholinesterase inhibitor and it shows cumulative effect is possible. This chemical may damage the nervous system with repeated exposure and resulting in convulsions, respiratory failure and may cause liver damage. Now we will discuss about methyl parathane. G. Schrader in 1951 and 1963 also synthesized a lot of other derivatives of parathane. Parathine methyl is of similar toxicity and its mode of action is somewhat same, but it is unstable if we compare it with parathine. Methyl parathine has low water solubility, it binds tightly to soil resulting in low leachability and has a short half-life in soils and on plants. Ethyl and methyl parathine are infrequently detected in groundwater sampling. Both are toxic to birds, fishes and aquatic invertebrates. Let us discuss the biological degradation of parathine. Parathine, ethyl parathine and methyl parathine have high acute toxicity in animal testing. In large doses, organophosphorus insecticides share a common mechanism of toxicity, that is inhibition of the activity of acetylcholinesterase enzyme in the nervous system. It results in excess acetylcholine at nerve terminals and producing acute symptoms such as nausea, vomiting, cholinergic effect, weakness, paralysis and seizures. Now we will discuss about malathion. This is the chemical structure of malathion. Its formula is C10H19O6PS2. Malathion is a non-systemic wide spectrum organophosphate insecticide. Malathion is one of the earliest organophosphate insecticide developed in 1950s and one of the largest selling organophosphorus insecticides in the world. Now we will discuss the properties of malathion. At room temperature, malathion is a yellow to deep brown liquid. It is often dissolved in hydrocarbon solvent before use. It has garlic-like odor. The quality grade can maintain its biological activity unchanged for approximately 2 years if stored unopened in a cool, shaded and well-aired place at 68 to 86 degree Fahrenheit. It is slightly soluble in water. Its solubility is 145 ppm at 20 degrees centigrade. Now we will discuss the uses of malathion. The range of versatile products is used for controlling a vast range of chewing and sucking insects in the fields of fruits, crops and vegetables. Other important uses of these products are like in grasshoppers and locust control, insect vector control, stored grain protection and control of pests in home, in garden and in also to control mosquitoes, flies, household insects, in farm and livestock parasites and for controlling head and body lice. Malathion may also be found in formulations with many other pesticides. Malathion is marketed as 99.6% technical grade liquid. It is available in formulations include wettable powder, that is 25% to 50% wet, emulsifiable concentrates, dust and aerosols. Now we will discuss the routes of entry of malathion. Malathion poisoning can be happen through ingestion, it can be through inhalation and by dermal contact. Let us discuss the harmful effects of malathion. The important route of exposure during formulation and uses is skin. So, prolonged contact for hours along with poor hygiene has resulted in irritation as well as symptoms listed under ingestion. And direct contact can lead to irritation and discomfort to eyes. Swallowing of malathion has caused severe poisoning and death, which include nausea, vomiting, headache, abdominal pain, diarrhea, difficulty in breathing, fall in blood pressure, muscle spasm, paralysis, loss of reflexes, convulsions and coma and death. If it enters through inhalation, so delayed pulmonary edema may occur after inhalation. Let us discuss the harmful effects of malathion after long term exposure. So, seizures are much more common in children than in adults. After long term exposure, death results from loss of consciousness, coma, excessive bronchial secretions, respiratory depression and cardiac irregularity. Commercial malathion products often contain impurities and hydrocarbon solvents such as xylene or toluene, which themselves can cause toxicity. The International Agency for Research on Cancer has determined that malathion is unclassifiable as to its carcinogenicity to humans. In animals, malathion induced liver carcinogenicity at doses that were considered excessive. The long term exposure also caused reproductive and development effects. Studies have been reported in which malathion induced transient testicular effects in rodents, and results from studies addressing that reproductive or developmental effects in humans are inconclusive. Prolonged daily contact with exposed area of skin has led to skin irritation and sensitization. The long term exposure of malathion may cause genetic changes. and birth defects have not been observed in humans exposed to malathion, but developmental effects may have been seen in offsprings of animals that ingested enough malathion while pregnant to cause health effects in the mother. And animal studies have shown that malathion can be transferred from a pregnant mother to the developing fetus and from a nursing mother to the newborns through the mother's milk. So, it transfers from a pregnant woman to the baby. Now, we will discuss the mechanism of action of organophosphorus pesticides, how these pesticides work. These pesticides kill insects by disrupting their brains and nervous systems. Unfortunately, these chemicals also can harm the brains and nervous system of animals and humans. The problem with organophosphates is that they affect an important neurotransmitter common to both insects as well as mammals. The neurotransmitter is acetylcholine. It is essential for nerve cells to be able to communicate with each other. And this acetylcholine released by one nerve cell initiates communication with another nerve cell, but that stimulation must eventually be stopped. To stop the communication, acetylcholine is removed from the area around the nerve cell by the malathion. So, these chemicals stop a key enzyme in the nervous system called cholinesterase from working, and enzyme acetylcholinesterase breaks down the acetylcholine. Organophosphates block this enzyme and disrupts the proper functioning of the nerve cell. Hence, these insecticides are called acetylcholinesterase inhibitors. Now, let us discuss how can we treat the organophosphate poisoning. We can treat it by first aid. We can start with first aid like decontamination, cardiorespiratory support and administration of the antidotes atropine and pralidoxim. In case of severe poisoning, diazepam, an anticonvulsant, should also be administered. Antidotes should be administered as prevention even if the diagnosis is in doubt. Now, we will discuss a new topic, that is bioremediation. What is this bioremediation? It is a waste management technique that involves the use of organism to remove or neutralize or detoxify the pollutants from a contaminated site. So, bioremediation is decontamination by microbes. Detoxification can also be done by plants, that is known as phytoremediation. It is the direct use of living green plants for removal, degradation or contaminant in soil, in sludges, sediments of surface water and groundwater also. Now, we will discuss the different types of bioremediation. There are two types of bioremediation. First one is in situ bioremediation and second is ex situ bioremediation. The in situ bioremediation techniques are applied to soil and groundwater at the site with minimal disturbance, while ex situ bioremediation techniques, in this method the soil and groundwater is first removed from the site via excavation or by pumping, and then bioremediation technique is applied. Now, we will discuss some different types of bioremediation. First one is bioaugmentation. As you can see in this slide, this is contaminants in soil, this is the polluted site, and bioaugmentation process, it is a process in which we externally added microbes. These are the microbes and they biologically degrade it or detoxify it, and then they form non-toxic end product. So, this is known as bioaugmentation. Another method is biostimulation. Biostimulation is just addition of nutrients. As you can see here, we are adding some nutrients to stimulate the population of microbes. If we add some nutrients, then the growth of the microbes will speed up. So, this is contaminated soil, the microbes digest it and they form non-toxic end product here. One more method is known as bioventing. As bioremediation is detoxification, so if we provide oxygen supply through water, through underground water like well, we can stimulate the growth of microbes. So, this is contaminated site again, the microbes digest this, they detoxify this and they make non-toxic end product. This is known as bioventing. Now, we will discuss the advantages of bioremediation. It is a natural and clean process and less equipments are needed. So, it is a less expensive process because there is not any requirement of equipment, and it is eco-friendly process because there is not any release of harmful gases. Now, we will discuss about the degradation of organophosphate poisoning. Organophosphorus pesticide form a major and most widely used group, accounting for more than 36% of the total world market. The organophosphorus pesticides are degraded by bacteria. That is very good point, that they can be degraded by bacteria such as Pseudomonas species, Agrobacter, Arthrobacter species, Flavobacterium species, fungi can also degrade it, Fusarium, Aspergillus niger, Actinomycetes, Streptomycetes and many more. Organophosphorus pesticides are highly toxic to humans, but positive point is that they do not remain in the environment for long periods of time. In this slide, you can see the degradation of malathion and parathine by microbes. This is known as microbial hydrolysis. This is parathine, and in presence of enzyme hydrolases, it can be degraded to paranitrophenol and ionic diethyl thiophosphate, and this is microbial hydrolysis. Malathion can also be degraded by Pseudomonas strain, and it degrades it into diethyl phosphorothioate and then dimethyl malate. Now, we will discuss about some genetically engineered bacteria. Biopesticides and microbial biopesticides, these are the subspecies and strains of Bacillus thuringiensis, which is known as BT. A native soil bacterium Stenotophomonas species, which is strain YC1, that produces methyl parathion hydrolase, that is MPH enzyme, and it was genetically engineered with the ICE nucleation protein from Pseudomonas syringe. The genetically engineered bacteria strain was able to degrade a mixture of 6 organophosphate pesticide, and it can degrade 0.2 mm each completely within 5 hours. So, viewers, this was organophosphorus pesticide. We discussed about the synthesis, their uses and their effects on human beings, their adverse effect on environments. After that, we discussed bioremediation, phytoremediation, and in next lecture, we are going to discuss one more family of pesticide, that is carbamates.